The recording begins. It's October 22nd, and this is Biology 105. Thank you all for being here. It's so lovely to see all of your smiling faces once again. Today, we are going to resume our discussion of cell communication. Now, I did a disservice to at least one of you, and maybe more, where I said on Monday, I'm not sure when the next exam is because I can't check on Canvas, but I know it's not next week. My most humble apologies, 100%. Exam 2's next week. I normally would have sent out an announcement on Monday morning like I always do, but yep, Canvas issue. I'll blame it on Canvas, though to be fair, I'm actually going to accept the majority of the blame on this one because I could have checked it on Sunday. But anyways, who knew that this change would happen? We'll try to just move forward past it. On Monday, you've got exam 2. There's a study guide posted for you. I did make an announcement yesterday about it. Here's what the exam 2 study topics document looks like. It's going to be exactly the same length, exactly the same sort of multiple choice structure that you saw in the previous exam. All right? So what, we did 25 questions, 3 points each. It's going to be 25 questions, 3 points each. Question?
Will it be an SI session or a practice exam?
There should be an SI section practice exam. I haven't heard from the SI leader yet. Is anybody here in SI who can? Okay, cool. We got a few people. Have you heard anything from your instructor?
I do remember she said, I do remember Finn said that he'll think of something, see what he can do, but he wasn't really thinking about that until we brought it up.
Totally. It's that point in the semester. This is when things fall through the cracks. Yes, at the back. Oh, because of an hour and a time limit and compensation, and because we have 3 midterms, there might not be one for exam 2. It's possible. There might be one for midterm 3 instead. So I can reach out to Finn and ask. I'm going to make a note here. This will get it done. Oh my gosh, if this pin would work. Oh, it's working. I just had to get it started. It's because my hands sweat. Anyways, I wrote Finn. That'll remind me, right? Yeah. I'll send an email out and check in on that. And if there is going to be an SI session, it'll be posted to the Canvas announcements. Awesome. Otherwise, focus on the study guide, the study topics thing. You can look these. I recommend if you have access to the textbook and any of these concepts you feel a little bit shaky on, look them up in the index of the textbook and find the pages that relate to it. Because I write my PowerPoints and my exams based on the textbook. So if you want to go straight to the source, Campbell Biology. It's my favorite textbook ever. And I've encountered a lot of different textbooks. It's great. Check it out. You need to borrow a copy? I've got a few extra in my office. Come to my office. I'll loan you a physical copy of the textbook. The library's got them. I've heard tell that there's free versions online, but I can't direct you to them. You have to find those on your own. But also don't do that for the recording. That's bad. I've told you how the textbook companies have your best interests at heart, right? They love all of us. They take a lot of, they do so much. They do so much for us. Anyways, I don't want to burn through all of our time because we are actually ahead of schedule, which is so rare. Let's go ahead and start getting into the material. Here we are, cell communication. There's a bacterial transformation quiz associated with your lab assignment. Hopefully you all saw that announcement about how somehow one of the lab assignments, when I adjusted a due date for an individual student, it deleted the mass due date, so I had to shift one assignment over. Just check your announcements. Everything should be in your to-do list if you still need to do it, okay? Check your to-do list on Canvas. That's that. Ready? Okay. Longer preamble than usual. I'm really excited, though, to get back into Week 9 material. Pick up where you left off. And yeah. Okay. So we have been talking about cell signaling. I talked about some different ways that signals could be transmitted. That includes the synaptic signaling, the gap junction, the autocrine, the paracrine, the endocrine signaling. All right, all of these different pathways just involve getting a signaling molecule to a cell. Now, sometimes that signaling molecule binds to a receptor on the surface of the cell and never actually enters the cell. It never crosses the phospholipid bilayer. So in order to get a response from the inside of the cell, the signal needs to change forms. And this is signal transduction. And there are many different signal transduction pathways. Sometimes a signaling molecule, if it's small and hydrophobic, in general, if it can cross a phospholipid bilayer, then it can diffuse all the way into the cell and elicit the response from inside of the cell more directly. So it interacts with an intracellular receptor rather than interacting with a cell membrane receptor, right? We talked about this on Monday. Any questions about those core concepts, please? Yes.
Maybe a dumb question, but I'm just not
There's no such thing.
Okay.
Sorry. That's what I'm supposed to say.
So the signal, like, can you go back a second? Sorry.
Absolutely.
So it bonds to this receptor, but then the signal changes shape inside of the like, because the signal is never inside. So how does it
Yeah.
change shape inside? Like, what's going on there?
Check it out. So the signaling molecule never enters, but the signal becomes something else. So in this example of the tyrosine receptor kinases, you have these external signaling molecules. They bind. They make this receptor molecule dimerize, where two identical pieces come together and form this now active dimer. And what this dimer does is it takes a bunch of phosphate groups and attaches them to these relay proteins. So now these relay proteins are activated and can elicit the response. So when I say that the cell changes form, I don't necessarily mean that the signaling molecule ever changes shape. I mean that the signal is this signal molecule present? And that's the signal that I'm talking about, is presence of the signaling molecule. But now, the signal has become activated relay proteins.
Okay.
Does that sort of make sense?
It causes the transformation of something else.
It causes something else to happen, and then the cell detects that internal change.
Okay. So I think that makes more sense.
So yeah, great. So signal transduction in general could be like, I heard something and then I wrote it down and I passed that to somebody. That signal was auditory and now it's visual. The signal has changed forms. The signal has transduced, or the signal has undergone transduction. Does that make sense a little more?
Yeah, no, that makes sense. Thank you.
Awesome, no problem. It doesn't mean, and I'll say it one last time, it does not mean that the initial signaling molecule undergoes any change. The signaling molecule is probably still there. Okay, solid. So if there are 3 steps to signaling here. 3 stages of cell signaling. 1, the signal gets received, reception. 2, the signal gets transduced, transduction. All right, changes form. 3, there's a response from inside of the cell. Usually for our teaching purposes, that response is going to be a change in gene expression or a change in regulation of a metabolic pathway, which could be mediated by a change in gene expression. Yes, please. What happens to the signal molecule? Solid. So the signaling molecule, it will stay bound to the receptor for some period of time, usually determined by the structure of the receptor and the structure of the signaling molecule, so it's different. And while it's bound to the receptor is activated and continues to bing, 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 trigger this response inside. Now, just like with not just like with an enzyme. Yeah, it is just like with an enzyme and an allosteric or a non-competitive inhibitor. This molecule won't stay bound forever. It will eventually pop off. And then that signaling molecule may be degraded. It's usually degraded once the response has been elicited. But there are a ton of examples of ways to regulate these responses by affecting how the signaling molecule is treated. So many psychoactive drugs, for example, they work by preventing naturally occurring signaling molecules from being broken down. So the signaling molecule just stays at a high concentration and continues to elicit the response. But the short answer to your question is they are usually broken down after a period of time so that the response stops. The transduced ones on the inside, they will be, it's a little tricky because most of the examples we're going to talk about deal with these molecules get phosphorylated and then they cause something else to change. Usually once they've carried out a given number of reactions, the phosphate group falls off or they change shape back. They don't usually get degraded. They usually get turned on or turned off. So it's a little bit different than the signaling molecule, which is usually destined for degradation, whereas the relay molecules are regulated like enzymes in a multi-step metabolic pathway. It's better to just turn them on or off so you don't have to keep remaking them. Does that make sense? Awesome. And there'll be other examples that doesn't fully fit with, but I think that's a good generalization that we can move forward with. Question, please.
So this process of transduction, does it also happen for the intercellular
Receptors?
Yeah.
Not usually. Not usually. At least not in the way that I'm going to teach it. So there could be a short signal transduction pathway. But usually with the intracellular receptors, what happens is the molecule makes it all the way into the nucleus, binds to its intracellular receptor, and now this receptor ligand complex, by the way, the ligand is the term we give for something that bonds to a receptor, or something which is bound by a receptor. It's its ligand. So the receptor ligand complex is the receptor bound to the signaling molecule, okay? So to step back and repeat this from the start, right? With the intracellular receptors, usually the signaling molecule diffuses all the way to the nucleus, and then the intracellular receptor ligand complex acts to directly turn on gene transcription. So it acts as something that we call a transcription factor, which we will talk about more when we talk about genetics and gene expression regulation. But think about it this way. In order for a gene to be expressed, it needs this. So as soon as this forms, the gene starts to be expressed. So there's not as much of a signal transduction pathway. Does that make sense? Awesome. Very cool. Additional questions about this stuff? Let's go through the steps in detail. Signal reception, okay? If the molecule is water soluble, it's probably not going to freely cross the phospholipid bilayer, all right? Therefore, the receptor is going to be a membrane receptor, all right? So far, so good? Three main categories. G protein coupled receptors. These are receptors that interact with something called a G protein, all right? The receptor tyrosine kinases. They're kinases. Kinases take phosphate groups off of ATP and attach them to something else. That root kine, like kinetic, like kinesiology, movement, energy. Kinases are a general class of molecules that take phosphate groups from ATP and attach them to things, usually activating the thing that they attach the phosphate group to, giving it more energy, right? So receptor tyrosine kinases. And the last one, which is the most mellow one, are the ion channel receptors. They're great. It's basically a channel protein in a membrane, right, that ions can cross, and they either open or they close. And if the ligand binds, they go boop, they open, and then all of the protons, all the ions can just flow. And then when the ligand's not there, they close. And there's no way for the ions to cross the phospholipid bilayer. Because remember, folks, ions don't cross phospholipid bilayers. right? Cool. Membranes are impermeable to ions. I love it. Hydrophobic molecules can pass through the cell membrane, through the phospholipid bilayer, and bind to intracellular receptors. We've already spoken about it. Any questions? Don't worry, we are going to talk about the 3 main types of membrane receptors in detail. Good, 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 good. I've scanned the room fully, and I see comprehension in all of your eyes. Very good. First, the G protein coupled receptors. All right. There's a receptor. There's an extracellular region that binds to its signaling molecule. There's an intracellular region that interacts with G proteins. Okay? When they receive their signal, all right, they're going to bind. I'll show you right here. When they receive their signal, they're going to bind to the G protein and attach GTP to it. Okay? Of course, you know, this process endergonic. Well, GTP production is binding the GTP here with GDP. Where does the energy come from? Don't worry about it. Stop thinking about things. Stop it. All right? Just absorb this stuff. No, be thinking about things all the time, of course. I'm just goofing. Just having a nice goof session. Okay. So check this out. We've got a G protein coupled receptor, okay? Here's an inactive G protein. When the G protein coupled receptor gets activated by binding its signaling molecule, it puts GTP onto the G protein. Now what's GTP? It's like ATP, except instead of the A nitrogenous base, it has the G nitrogenous base. So it can still serve as an energy source because it has that high energy terminal phosphate group. All right, additional details that you don't need to be concerned too much with, but it should make you think, like, oh, there can be GTP? That's kind of cool. Yeah, they totally can. Okay, check it out. Once this G protein has GTP bound to it, now it's activated. And now it's going to go interact with something else, okay? In this example, it's activating an enzyme, giving that enzyme the power to elicit a cellular response. Maybe that's by breaking something down. Maybe that's by activating a metabolic pathway that then is going to regulate gene expression. There's all sorts of things that could be happening here, okay? Maybe this is just directly affecting a metabolic pathway. You know, you detect that there's something that you want to break down outside, so you just activate the pathway to make the enzymes that are going to break down the path, that are going to break down the thing that is outside. Reasonable enough? Okay, solid. So G protein coupled receptors interact with things called G proteins that they activate by attaching GTP to them. That's basically all you need to know about this class of receptors. But trust me, when you start looking, if you ever start like reading papers about cell biology, you're going to be stoked that you know about these 3 types of membrane receptors because they come up all the dang time. Okay? Hey, look, there's a bunch of them. Don't worry about it. Good deal. Oh, am I already going to talk about phosphorylation cascades? No, I'm not. Okay, so the G protein coupled receptors, to be kind of a jerk about it, they're not actually making GTP. They are attaching GTP to a G protein. All right? So that's very different. So while there is movement of a molecule that contains a phosphate group that has a bunch of energy in its terminal bond or in its bond, it's different from this class of enzymes which we're now going to start talking about. This class of enzymes being the kinases. All right? Kinases specifically, they take the phosphate group from ATP and they attach it onto something. They phosphorylate something, all right? So kinases phosphorylate things. And when something gets phosphorylated, well, now it's got more energy. it could go on and carry out a reaction. Remember our discussion of phosphorylated intermediates and how energy from ATP can be used to do work by temporarily phosphorylating a molecule and raising its energy level so then it can do something else. It's all very vague, but that's going to be okay, I hope, for us. These kinase enzymes, right? They're incredibly important because they take phosphate groups and attach them to things. They take phosphate groups from ATP, attach them to things, give those things energy to do more work. Cool? Or turn on the enzyme or turn on the protein, allow it to carry out some different function. Their mode of action is move the phosphate group. Okay, that's every kinase enzyme. We cool so far? A lot of these receptors operate by having a kinase domain. All right? So they receive a signal, and then they go, time to be an active kinase. I'm going to transfer phosphate groups. That is not how the G protein coupled receptors work. I'm emphasizing that again. But the receptor tyrosine kinases do work in this way, okay? Or the RTKs. So, RTKs have a weird structure where there's two more or less identical pieces, and the RTK only becomes an active kinase when both of them get activated by a signaling molecule, and then they snap together, and then they can start to transfer phosphate groups and activate things. But here's the thing, right? You get a signal, boom, and then blam, blam, blam. See how that's activating or amplifying my signal inside? So we're going to talk about how these can set off phosphorylation cascades and amplify signals. Alrighty. So anyways, it's not especially useful to make generalizations about what the different types of receptors do, because these are three broad categories of receptor types, and each one can elicit all different types of cellular responses. That said, receptor tyrosine kinases have a natural capacity for amplifying signals, and so if you're trying to get growth factors to do something inside a cell, which is usually pretty broad spectrum response, where like everything needs to start dividing and growing, it's time to grow. RTKs are commonly involved in interacting with growth factors to elicit the cell growth, cell division response. And so as you can see right here, if you have an abnormally functioning RTK, maybe you've got, it can lead to some sort of cancer. Question?
Why is it important that these kinases attach to tyrosines?
It is only it's not important for our purposes. It's important for their naming. You know, they get their name because they have these tyrosines. Okay, these tyrosine groups. It's the active region of the protein. It's not something, while it is something specific about the tyrosine, let me just show you. While it is something specific about the tyrosine structure that allows this interaction, this figure does not go in depth enough into the structure for us to understand that. Does that make sense at all? It's a biochemistry thing, is the disappointing answer for this. But it's important for the naming convention of the active regions of this receptor have tyrosines in them. Yeah. I remember when I said, I think on Monday, I was like, this is one of the less satisfying sections of this course because so much of it has to be just like, yeah, this is a class of receptors and here's how they work and don't worry about it. You know, it's never satisfying for me to give that response. But here's something you should worry about with the receptor tyrosine kinases. Check it out. There are 3 active domains, 3 active kinase regions on each component of the receptor tyrosine kinase dimer. That means that when this thing dimerizes and becomes activated, there are now 6 operating 6 operating kinase domains, okay? They've all become activated here. That means each one could interact with a different relay protein. So you could elicit 6 separate responses. Or you could interact with 6 of the same relay protein. So an initial signal of 2 signaling molecules becomes, boom, 6 signaling molecules inside. So it can make a pathway branch, or it can make a pathway get amplified. So you can elicit from a single signaling molecule, you can elicit a variety of responses, or you can elicit a bigger response, you see? So that's one thing about these receptor tyrosine kinases that's, pretty useful to know about. An important thing about their structure. Okay, how do we feel about kinases? I'm going to talk more about them, but I want to get it all ironed out now. Yes, please.
I guess I'm just kind of confused because you said kinases are different from GTPs in the way they function, but then RTKs are the same as GTPs but are different from kinases. But
RTKs are not the same as G protein coupled receptors.
yes, but you said they operate.
GTP acts a little bit, it's tricky. So GTP acts like ATP a little bit in that it has energy in its terminal phosphate group. Kinases specifically take a phosphate group off of ATP and then attach it on to something else.
Okay, so kinases take a phosphate off of ATP, so they use ATP, but G proteins.
G protein coupled receptors.
Yes.
And then G proteins. Okay. So G protein coupled no problem. So it's 2 pieces here.
Okay.
The G protein coupled receptor and then the G protein. The G protein coupled receptor receives the signal and then attaches GTP to the G protein, activating it. And then the G protein goes off and does something else and elicits a response. Now it's been activated.
So in this case, the energy is being attached to what it's going to activate, but in the other case, energy is being taken to cause activation.
In every case, energy is taken to cause activation, but you're just doing it, you use some energy to activate the thing. It took energy to activate the G protein, but then there's more energy available to elicit the response. These things are going from very, very high energy to lower energy states. It's a little bit tricky to get the energetics piece in here as well. It's good to try to parse everything, get it all to operate together, but we don't really have all the pieces to allow for that yet.
Yeah.
There's the reason why it won't come together in your brain is because there's connection stuff that isn't there yet.
Yeah.
So that's totally, yeah. Oh no, it's not only on your side, we're gonna get there.
Okay.
Yeah,
Great.
yeah, and then we'll visit it again and see if we got to the point where all the connections get made. Awesome, no problem. Yes, please.
Yeah, so how much of our cellular energy comes from GTP versus ATP?
Oh, I have no freaking idea. Yeah, I will tell you right now. I'm going to repeat for the video question. How much of our cellular energy comes from ATP versus GTP? I can tell you that much more, the majority comes from ATP. But GTP can be used as well, and it does contain energy as well. But think about it this way, right? There are all of these enzymes that are involved in transferring energy from ATP to other things. Those things don't work with GTP. So GTP is going to be produced for specific functions to work with specific enzymes, and I can tell you that it is not a primary source of energy, even though its structure allows it to function as a source of energy in a way similar to ATP. The issue is that there aren't enzymes that work with it. So you know what? I kind of had an idea of how to answer your question. I just don't have a number to give you. Yeah. Satisfying enough? Awesome. All right, cool. My clicker. Yeah, I have to press the buttons. Okay, so what do you need to know about the receptor tyrosine kinases? The receptor tyrosine kinases, they are kinases. They transfer phosphate groups from ATP onto other things. All right, you should know that there's 2 subunits, and when they receive their signal, they dimerize, and then they phosphorylate. They become active as kinases. Why do I focus on it at all? Because kinases are incredibly important. So I like to talk about the receptor tyrosine kinases. I've gone back and forth and spoken to other faculty and tenured faculty and all those sorts of people here, about like, how much of this should I be covering? And there's a variety of opinions, and I've settled on this depth as, I think, serviceable for the rest of you, for your further education. Go to Cell Bio. You'll get a lot more about these signaling things, and they are very interesting. Okay, the last class, ligand-gated ion channels. I save them for the end because they're the fun ones. They're the simple ones. It's easy to visualize. All right, check it out. You've got a bunch of ions sequestered to one side of a membrane. The ions can't freely cross. Signaling molecule comes by, interacts with the ligand-gated ion channel receptor. So when the signaling molecule, the ligand, binds to the receptor, the gate opens. And when the gate opens, the ions diffuse through the channel. And then the inside of the cell detects that there are more ions. And that is enough to start the response of the cell. This is pretty fast, super useful for when you're trying to get like a defensive response in plant cells. They'll just open these channels and let different ions, usually calcium, just flood into the cell. And the cell goes, we're under attack. Let's start becoming really gross. And sometimes the cell will just make a bunch of poisons and unpleasant things that may kill the cell, but it knows it's about to get munched anyways, so, it gives itself up. Anyways, pretty straightforward ligand-gated ion channel receptors, right? Right? Open up the gate, allow the ions to diffuse. Okay, cool. Intracellular receptors, all right? So the intracellular receptors, they could be in the cytosol or they could be in the nucleus. But oftentimes these are going to be interacting with hormones, steroid hormones, thyroid hormones, which come from their origin in that gland, all right? But either way, these hormones are small and they are hydrophobic. Therefore, they can cross the phospholipid bilayers and reach intracellular receptors. Oftentimes, when the hormone binds to its receptor, it acts as a transcription factor, allowing transcription of genetic material. It allows the expression of a gene that otherwise would not be expressed. So that's our standard intracellular receptor function and intracellular receptor mode of action. That's the other way of saying function, but I couldn't come up with a word on the fly. That's okay. No one even noticed, because I played it off. So cool. So cool. Any questions about intracellular receptors? We've been talking about them. I've told you so many times how much I care about the permeability of membranes to things, so I'm hoping that this is pretty cemented in your brain. A question?
Could you expand a little bit on what transcription means?
Absolutely. That's so great. So we haven't reached our genetics portion of the course yet, and that means that we haven't really had a chance to talk about the central dogma. Somebody took the spiral notebook. Does somebody have a blank piece of paper? Thank you. Awesome.
Pencil too.
Nope, I got a pen I can write with. All right, thank you. What am I doing here? I'm writing. Central dogma. With a name like central dogma, you better believe that it's important. All right? It is central to biology, and it is dogmatically adhered to. A dogma is something that you just believe and you don't question. So don't question anything I'm about to write for you. It's the central dogma, okay? Here it is. There is this DNA, all right? The DNA gets read and is used to direct RNA. the RNA is then read and is used to direct a protein or synthesis of a protein. So the DNA encodes RNA structure, the RNA encodes protein structure. So far, so good? The process wherein DNA is read and used to direct the synthesis of RNA is transcription. The process wherein RNA is read and is used to direct the synthesis of a protein is called translation. So transcription factors, what they do is they encourage transcription of a specific sequence of DNA into RNA, right? And that means that now that RNA is there, and it can be translated into a protein, okay? Now, there are other types of RNA. Only mRNA actually gets, or messenger RNA, actually gets translated into a protein. But there are other types of RNA, like tRNA, or rRNA, ribosomal RNA, or siRNA. Okay, there's all these other different types of RNA that can be produced via transcription, but never actually get translated. But the central dogma is just this, okay? DNA gets transcribed into RNA, which gets translated into proteins. And I'll say it again, it is read and is used to direct the synthesis of, because the DNA doesn't get converted into the RNA and the RNA doesn't get converted into a protein. It encodes the sequence of the RNA and the sequence of the protein. Yes, please, question.
So RNA basically acts like a blueprint?
Yeah, it's like I used to do this analogy of there's a castle, and I get to be the king because I'm the teacher, you know. And I live in the castle, right? And I've got the ancient blueprints before me, and I know how to make, I don't know, a magical potion. Let's just have fun with it. I've got the tome. I'm not letting anybody see it. I'm in my little nucleus, and nobody better bother me except my royal advisor, who comes into the room and goes and whispers into my ear and says, "Master," you know, and talks to me like that. And so what I do is I open the royal tome and I say, "Be gone," and I'm like sick and I don't look so good, and I eat a tomato and it's gross. From the movie, remember? He eats the tomato. Okay, so then I take that DNA, the core instructions, and I say, "You can't look at this," and I write down the instructions in a temporary form that is crappy. It's on a piece of parchment that could totally degrade. It's less stable than the tome, and I give it to my evil advisor. He says, "Yes, good." And I take the RNA, and then I leave the nucleus, and I walk down into the village, and I say, "Disgusting," and I throw the little message before the serfs, and I say, "Make the potion." And those are the ribosomes. I'm the king, just period. I'm not an analogy for anything. I'm just the king, all right? And the tome was the DNA. This here, the RNA transcript, was a little temporary message that had the same information on it. And then whoever gets that message has to do the work of mixing the chemicals together and making the potion. And that's the ribosome, and they make the protein. How's that? Cool. Huh? It's gotta be. I'm not. No, it's freaking. It's Lord of the Rings. Because I said the tomato. The guy eats the tomato. That was a flawless worm tongue. I swear. Thank you. I had another calling. I had other dreams, you know. But you know, those dreams don't always get realized, and that's okay. Yeah. Everything's fine. Don't worry about me. Honestly, I have the best job in the world because I have the best students in the world. I am not complaining. Thank you all. Okay, so that's the central dogma, all from the question of what is transcription. But now we understand what a transcription factor is, yes? Yes. Okay, cool. The transcription factor is somebody coming to me, the king, and saying, hey, we really need some of those potions. And then that allows for the creation of the transcript. Awesome. Hey, check this out. Intracellular receptors, this is actually more accurately the molecules that interact with intracellular receptors. Check out their structure, like testosterone, estradiol, cortisol, vitamin D3, all right? They've all got these long hydrocarbon ring structures, which, as you know from our biological macromolecules talk, these are all hydrophobic, all right? It's hydrogen-carbon, hydrogen-carbon, non-polar covalent bonds, hydrophobic molecules. So you can see how all of these could theoretically diffuse across a phospholipid bilayer. Cool? Awesome. Oh yeah, endocrine disruptors, so things like DDT, okay, BPA. There are some molecules that that are similar in structure, and they can signal a similar response to those hormones. They're like hormone mimics. Like BPA was the thing that everybody was concerned about in water bottles for so long, and now people just talk about microplastics, and I feel like we've kind of lost sight of the BPA question, but you know, they don't put it in the water bottles anymore, and there's no other ways that you get exposed to plastics, so don't worry about it. Just don't stress. All right? There's a lot of really interesting data out there about how much BPA has been absorbed into your body from sitting on plastic toilet seats. Yeah, now that's a new thing that you can be worried about. Just put it out of your mind, you'll be fine. You're so welcome. All right, so there's nothing key really about these except, look, these look like these and they can elicit the same responses. Okay, cool. Signal transduction. So all of that so far was signal reception stuff. Receptors, right? Bless you. We've got the G protein coupled receptors, the ligand-gated ion channels, and the receptor tyrosine kinases, right? The three different classes. Now we're going to, oh, wow. Well, it looks like on Friday, we will be talking specifically about the signal transduction step with a lot of focus on protein phosphorylation and dephosphorylation, including kinase enzymes, and the opposite of the kinase, the phosphatase. Thank you all for being a part of Bio 105, because you can have a class without me, but I can't have a class without you. See you all on Friday.
Are we going to get to week 10 and week 10?